Welcome, everyone. Thank you for being here today. Another one of our wonderful webinars. I am Isabel, CEO of Renegade Research and Project Director for Remission Biome. I am an ME/CFS patient of 43 years, and I am also a systems engineer, board-certified health coach, and have extensive experience in the pharmaceutical sector. If this is your first time with us, we are very happy to have you here. We are a volunteer-run nonprofit organization focusing on decentralized research, clinical education, and practical advocacy for infection-associated chronic illnesses, with an emphasis in ME/CFS, regardless of trigger. Today, we are focusing on our project, Remission Biome. Again, we are run by an amazing team of volunteers, the majority who have personal experience in this condition. Next slide, please. And today, we would like to give very special thanks to the data team that has put all of this together and will be shared details of the data. On the agenda, we have myself right now to do an introduction to the Remission Biome Project. The data analysis team, starting with Mike, will do the overview, the Bearable and wearables that we use to measure, what we have in terms of symptom clustering, preliminary Biomesight results, genetic patterns in our cohort, and the surveys that we have done, as well as the summary at the end. So next slide, please. Again, we would like to thank the data team, and we would like to give a special shout out to the data team and everyone that is here, starting with Mike, Lisa, Peter, Nick, Kat, Josh, Sam, Tess, Elly, Sam, Tara, and Thomas. So thank you, everybody, for all of the efforts and everything that you have done to put this together. So what is Remission Biome? Remission Biome is an experimental project with a medically supervised protocol that is designed to assist patients with ME/CFS and long COVID in potentially achieving a higher baseline in function and improving quality of life. The updated protocol is actually going to be released in our clinicians roundtable with Dr. Hutton coming up on February 7th. The protocol originated from patients reporting transient remission events, which we will call TREs during this presentation, where all of the ME symptoms resolved after taking antibiotics prescribed. Most of the antibiotics were amoxiclav, and they were prescribed for various infections. In a remission, symptoms that aren't attributable to ME/CFS do not necessarily resolve. So for example, if someone has back pain from a bulging disc, a transient remission event may vary in duration from minutes to months, and in many cases lead to improvements in baseline function for various periods of time. So we're going to be looking at the timeline that in which this project has taken place. So in September of 2022, a discussion started on Twitter between patients that had experienced previous remission events and baseline increases after taking antibiotics. So out of this, a pilot self-experiment was conducted in March of 2023 with 3 participants, recreating some of the supplements that those participants were taking at the time of prior remission events. And 2 of these participants also conducted extensive testing to understand the mechanisms. For this pilot experiment, the goal was to see if the transient remission events could be recreated. And 2 out of the 3 experienced multiple transient remission events while taking the antibiotics and subsequent baseline increases that lasted for many months. I was one of the participants, and my baseline decreased after an acute respiratory infection 9 months later. But even after multiple infections, including COVID, it has not decreased anywhere near back to the level before the experiment, before the pilot self-experiment. We share this success with the ME and long COVID communities on Twitter, now X, and others in the community reached out between April and July of 2023 because they were being prescribed amoxiclav for, again, several infections. We share the components of the protocol, and they implemented as many as possible of those components for up to 1 week before starting their prescribed antibiotics. They recorded their experiences of transient remission events and baseline increases for those who had them. So Tess will be sharing more on this later, but we call this group of people the 1.5. In July of 2023, we put out a call to the community to formally organize a larger group of self-experimenters and aggregate all of the N-on-1s with their provider supervision. Over 500 people applied, and in July 2023, we admitted a group of 50 participants from all over the world and from the entire spectrum of severity to form what we have called the Renegade 50. So the data that we're sharing today is focused largely on the transient remission events with some additional insights from the Renegade 50. However, what we started as looking to recreate the transient remission events as a window into mechanisms of potentially reversing ME has evolved. Our objectives have expanded. In the R50, we have focused the immediate work more towards baseline increases because quality of life improvement now is just as important as understanding how to reverse this condition in the future. We now know that transient remission events can be recreated. It has happened multiple times across multiple groups. We are working to understand why it happens in some and why it doesn't in others. We do know that having TREs is not necessary to experience baseline increases on the protocol, but still need to understand if TREs influence the level of duration of such increases. To understand what happens to our health baseline long-term, we need to better understand what happens physiologically during those TREs. Our current main objective on the protocol is to achieve those baseline increases with or without the transient remission events and with or without amoxiclav as part of the protocol. The project has expanded over a much longer period of time than we originally thought or intended. We have spent this time understanding responses, collecting data, troubleshooting individually and collectively, and overcoming obstacles, whether that's logistics, funding, limited spoons, so that future cohorts and our other research projects can benefit from everything we have and continue to learn doing this decentralized research across the entire spectrum of severity. Next slide, please. So where are we now? As of right now, the protocol consists of 6 stages. Originally, we only had 3 in the pilot. So participants undergo these stages under supervision from their healthcare providers. So I'll spend a little bit of time on stage 1 for the most part, which consists on extensive safety testing. There are standard tests as well as optional ones and individualized tests based on the profile of the participant. This stage allows participants to work really closely with their providers and address underlying issues with a lot more precision. This stage was added based on observations in the early stages of the project, especially after the first 2 participants went through the antibiotic phase. There are several factors identified as potential barriers to success on the protocol: co-infections, especially Lyme or mold, frequent acute infections pointing towards more severe immune dysfunction, and severe mechanical issues, as well as hormone disruptions all affect the effectiveness of the protocol and how people fare longer term. More recently, Kat will be talking about this later, also how genetic variants influence the success of the intervention. All of these factors need to be worked together from a systems perspective, not as an isolated factors or even the sum of their parts. Once we have determined how these factors play together and if they can sufficiently be overcome within the context of this project, we proceed to the next stages. So stage 2 is a holding pattern in order to have that baseline before going into the core of the protocol. Stage 3 is what we called before the prep phase, which is a number of interventions to prepare the system for the antibiotic phase, which is stage 4. But then again, we also have a track that does not include an antibiotic. Stage 5 is to reseed the microbiome and restabilize the gut-brain axis. And finally, stage 6 to further stabilize the entire system and the overall baseline in order to continue a trajectory of improvement that is individualized and can be sustained. And I will pass it out to Mike.
Thanks, Isabel. I'm Mike from San Diego, diagnosed with long COVID, ME/CFS, and tethered cord. I'm the team lead for the data analysis workstream for Renegade research. The Remission Biome Project is a patient-led Renegade-style self-experiment. Most of us on the data analysis team are dealing with chronic illnesses ourselves. And probably our biggest challenge, as we say in the chronic illness community, is that we have limited spoons. Our energy levels can fluctuate dramatically, and that can make it hard for us to make progress. So though I'm having a good day today, I've been on quite a turbulent journey since the COVID era began. Back in 2020, before this project ever existed, I started having profound and debilitating symptoms and rapid fluctuations in mood, sleep, and energy. Now, my background is in IT and I love data, so I started using a mobile app called Bearable, which allows you to log and quantify symptoms, medications, mood, and lots more. By doing so, I could visually see what was happening to me during a chaotic time, and that helped me identify clues, track my progress, and hopefully give me insights for making data-driven decisions about my own health. So once Remission Biome started, I volunteered to help others capture their health journey into the Bearable app as well. Now, all participants in the Remission Biome project use Bearable every day to log their data, and that data can then be exported and gathered centrally for analysis. So as we began to make plans for the Remission Biome project, it became clear that we had an opportunity to create something truly unique. And we look at data in two ways. First, on the left, individuals can look at their own data for personalized insights. And using this data can help guide each participant on their own health journey. You might say that's looking at n equals 1. Second, on the right, we can look at this data from a group perspective. So we can see how the protocol is working and refine it over time. This group data can help us tell the story of Remission Biome, just like this presentation today. And all of this ultimately helps us to move science forward and to help people improve their health. So beyond data entered into the Bearable app, we also capture objective data with an array of wearable devices, labs, tests, surveys, and more. Let's dive in just for a minute to see what some of this looks like. This is our framework of data sources, which combined give us a very, very rich data set. On the left side in blue is participant reported data. This includes a daily log of every medication, supplement, and probiotic that each participant takes. People track mood fluctuations and symptoms on a numerical scale. And from these logs, we can calculate daily scores, which can be graphed and tracked over time. We're also using a tool called BrainCheck in the standard scoring systems Bell Score and FunCap at regular intervals, for example. On the right side in purple is hard objective data, including wearables like Oura Ring and Apple Watch. Over time, we can observe changes to things like resting heart rate variability, sleep metrics, and lots more. And in the last column, you can see we capture a variety of labs and tests, including Biomesight stool samples for gut bacteria, the Cyrex mucosal test, the organic acids test, and others as well. Data from all of the sources that you see here on the screen is exported to a CSV or PDF formatted file, and all of these files are gathered, anonymized, and stored in the GitHub platform, giving us thousands of data points on each participant. So this data is deep. In other words, it's gathered in very great detail. It's diverse because we're drawing from a lot of different sources, and it is longitudinal, tracked over months or even years, which means that this project is really, really quite unique. We believe that truly incredible insights can be gleaned by connecting all of these data sources together. In our data store, every file is indexed by a participant ID, and nearly every data point has a date stamp and quite often a timestamp. Because of these two indices, we can fuse data in very interesting and whole new ways. We can combine wearable, bearable, and gut bacteria samples, for example, before and after going through the protocol. So we can see how symptoms and heart rate might correlate to treatments, interventions, and time. What you're about to see today is a mix of early results, ideas, and hypotheses. Some of these are not fully fleshed out just yet, but we hope these are the seeds of great discoveries. And with that, I'll hand it over to Elly.
Thank you, Mike. My name's Elly Brocious. I have had a diagnosis of ME/CFS for 32 years, although lots of sickness, viruses, sensitivities before that. I am in stage 1 and 3, taking products from both of those parts of the protocol as I work to clear things like mold and heavy metals and plastics from my system. Apparently my genetics show that I have a terrible time clearing toxins, and so I'm working hard on that now and looking forward to taking the interventions that come later. My jobs within Remission Biome are to be a small group protocol leader and listen to people's experiences and help them with their questions. And also I'm on the data analysis team, and I look at the Bearable files when they first come in. So on this slide, you'll see a spreadsheet looking thing, a CSV file, comma separated values. Our people do not type in their stuff like this, right? The little app where you enter your data as much as you can. Daily is great, but a lot of our people have to take breaks and only do maybe 1 item a day if possible. But we often have people doing much more than that. So I look at this file with my eyes on the actual data, and I run some little app scripts on them. And I try to make sure that we have sort of a consistent standard level of data of the required items that people are doing, symptoms, meds and supplements, mood, which stands for overall health in our case. Mood is a built-in thing to Bearable, but because it's so handy and prevalent, we use it for overall health. So I look at these things and I make sure, not just the required things, but I also see if some people do extra items, because that can be quite fascinating as well. Okay, next slide. So I check the data, look for some standardization across so we can compare and contrast. I look for patterns in individuals so that I can follow up with questions to them. But what we need to realize here is on February 12th last year, we noticed that we needed to adjust things. We were just asking people up to that point to track their remission biome treatments and interventions and their overall health. But we said, we need some extra data here, because we are all complex and we're all taking lots of things and we all have lots of other symptoms. So please, we changed it after that to collect all your symptoms, all your meds and supplements, and do 3 ratings of cognitive function, physical function, and this overall health. And we more clearly defined in Bearable what the terms mild, moderate, and severe stand for, because people were interpreting them a little bit differently. So we had to harmonize that. standardize that. What I've noticed is there's huge variations. I mean, it was going to be obvious, right? We are all very different, experiencing different symptoms at different phases of our illness. But we're trying to make it so it makes sense, that we can see signals, so that we can learn from what interventions we're doing. And it's amazing to me that some people can track over 100 things a day. Sometimes it's because they have their wearables connected. Sometimes it's because they take a lot of medications multiple times a day, and they can hit copy last entry to copy everything from yesterday's medicine over to today. So it's not too labor intensive. And some people track 7 or 8 moods a day, overall health, because it swings that much for them with their circadian rhythms. And other people are only able to capture maybe 1 mood a day, or they only need to because they're so stable. Nothing seems to change for them. Next slide, please. Okay, each column of colored boxes here represents one of the TRE5, we're calling them, the people who had transient remission events. And all I did here was look at the required data, which was represented by the 5 colors, and see what was there, how many, not what quality, not telling me any ratings of any kind, but just how much is there. So the orange represents cognitive function. And in the early data, we didn't have people tracking that, so you'll see some zeros. The purple is physical function. Again, we added that later on February 12th, 2024. But blue is our medications and supplements. How many entries per day? Or I mean, in this case, it's a total one. Total for the variable file that we have. Red is symptoms, how many symptoms they're tracking. And green is how many moods they're able to capture. So it's interesting to me, just looking at a quantity level, that the people who are tracking way more medications have fewer symptoms that they're tracking. Does that mean anything? We don't know, because they could have 3 symptoms that they're tracking daily that really are upsetting to their quality of life. You can't tell anything qualitatively from this, but it's just interesting how much it varies in just these 5 people to me. So if you look at the bottom, it tells you the total entry points we have, even for the smallest data set. On the left here, we have almost 2,000 points over 122 days. And in the middle, we have 85,000 points for 1,500 days. So we just let people be themselves and track as much as they want in between when they're going to take the interventions, but closer to when they take the interventions, we ask for much more careful tracking. Next slide, please. One of the things that Bearable lets you do is put in notes, and I wanted to share some encouraging ones that I've read. These are very individualized, obviously, and your mileage will vary. It's not just may vary. So here's some things I've read. The interventions, the person talking about, had a big positive impact on my stress reduction, and I had an HRV increase. That's exciting. This person, in order to get an MECFS person to type something, they have to probably be excited about it, right? The next person, this is 1,000% unusual. I never poop first thing in the morning. I've never pooped regular poop. It is always a seesaw of complete constipation or liquid diarrhea. And then it goes on for many days, and the person comes back and wrote, update, my morning normal BM type 4, which is the perfect snake BM on a Bristol scale, it continued for 17 days. This is from adding interventions from Remission Biome. I've checked with the person that did not continue because their doctor prescribed a medication that caused constipation, but they are still 1 year later excited that this happened to them, and they feel hopeful that this can happen again with more intervention. We see a number of quotes like this one: muscle tension I've held in my shoulders all my life is gradually melting away with these stacked interventions. Another person wrote, shoulders extra relaxed from 2 PEA yesterday. Another person took 3rd dose of amoxiclav earlier. I had a late afternoon remission event, went out to dinner, and had an MCAS reaction. Didn't realize that blurred vision had gone away until it came back. And I also realized I had less urinary frequency until it came back. So this is something that's exciting. When you feel better, you don't even notice what's going on. You just notice you feel a lot better. And then you can start, things start coming back, you can really tease apart symptoms. So I'm glad people are capturing these in their notes. It gives us direction in the data team, and it helps the coaches advise patients more and understand their experience. Next slide. This is my final slide. This is a real data. And this is probably the best example I can show you of a person who's on the left side is establishing their baseline. Then where the green line is, they've added the amoxiclav, the antibiotic intervention, and then they have some cycling, and they even hit 10 on mood. 10 is your maximum possible mood where you feel healthy, you have no ME/CFS symptoms, you feel good. So they had 3 of those over the next few days. Their baseline is slowly creeping up, lots more 8s and 9s, and then look at that stabilization on the right, just day after day of feeling okay. The person, cannot maintain this if they're out in the real world and stress is happening to you and new infections happen, but they can get back there because now they have new ways of reestablishing better feeling. Okay. After I am done with looking at Bearable data at first glance, then it gets turned over to the people who are able to do more complex math on it. So the next person is going to speak is Thomas, and he'll tell you some more about it.
Hi, I'm Thomas. I'm a postdoctoral scientist at the Vermont Complex Systems Institute. I don't have ME/CFS. I do have a handful of more mild long COVID symptoms, and I'm a volunteer with the Remission Biome Project, hoping to use some of this Bearable data to understand the structure of ME/CFS as an illness. So we all know that people who have ME can suffer from a really dizzying variety of symptoms that affect almost every system of the body: neurological, circulatory, digestive, everything is in play. And what we would like to do is we would like to use these symptoms to try to understand what systems are dysfunctional and in what ways. And so the very simplest analysis that I can go through here is looking at how symptoms co-occur. So on a given day, you might have 2 different symptoms, which we'll call X and Y, and we say that they occur with some probability, right? There's some probability that you have a migraine, there's some probability that you have a MCAS reaction or something like that. And then what we can look is we can say in the Bearable data, if these 2 symptoms co-occur more often than you would expect by chance, then they would share a common cause, right? If you have nausea and bloating always occurring together, that would suggest that there is something wrong, perhaps with your microbiome, that's being reflected in 2 different symptoms. In contrast, if the 2 symptoms co-occur less frequently than you would expect by chance, then we expect that those symptoms are being driven by 2 different systems that are interacting in some complex and non-trivial way. And so if I could get the next slide, please. So what we can do is for each person, and then also for the whole population of all the Renegade 50, I can go through and I can compute for every pair of symptoms, this measure of whether they are co-occurring more frequently by chance or less frequently by chance. And I represent them on these big matrices where every row is a different symptom, every column, is the same symptom. And then if the color of the box indicated by that row and column is red, those symptoms co-occur more frequently than you would expect by chance. If it's blue, those symptoms occur less frequently than you would expect by chance. And what's really cool is we can do this both for individuals and for the population level. So for this individual, this would be person 30 in the Renegade 50 cohort, we can see that, tremors, sympathetic overdrive, hives, and low blood pressure, they all tend to co-occur together. We can group them into this big box. And so this suggests that maybe there is some symptom that some underlying pathology that is driving all of these symptoms, which is different from, this other set of symptoms here, which forms a different community in this large symptom network. We can also look at the whole population level, and we can look and see, one of these communities is abdominal pain, nausea, flatulence, bloating, low appetite, and diarrhea. This might suggest some kind of SIBO-like or other microbiome-related pathology, whereas there are other clusters down here that suggest some kind of central nervous system issue or spinal issue. And the hope is that, if we have enough data from individuals, we can actually use this to help tailor their particular treatments by understanding what the underlying sort of unobservable pathologies are that are manifested by symptoms and recorded in Bearable. Can I get another slide, please? Okay, so that's one of the projects that we're working on the data team. The second project I wanted to talk about is this Transient Remission Event case study series that we're working on. And so Isabel made reference to this in her introduction. We're talking about 5 individuals who successfully had transient remission events induced as part of the pilot studies, who were also collecting lots of wearable and bearable data at the time. So we can see here there is plots of mood and energy for one person. And so if we compare the 90 prior days before they started taking the antibiotic with their mood and energy during the antibiotic, we can see that there is a small but statistically significant increase in mood and energy. And so what this tells us is that we've captured these transient remission events in data. It's no longer just self-reports or anecdotes, but we can actually see these things in data. And can I get the next slide, please? And the hope is that we can use this to start drafting a peer-reviewed scientific manuscript that will introduce this concept to the scientific literature. And so some takeaways that have already started to come out of this analysis is that, beyond just that these transient remission events can happen, they can even happen to people who have been extremely sick for months or even years. People who have been very disabled for a long time can rapidly achieve dramatic increases in capacity and quality of life when the right interventions are done. Unfortunately, figuring out what the right intervention is proving to be complicated because different people respond to different antibiotics. Antibiotic A might trigger a TRE in one person, but not another person. Antibiotic B might work for that person, but not the original person. And some people have taken antibiotics multiple times, and they don't always get the transient remission event. So we don't quite know how to reliably induce these yet. And of course, they're transient, so they don't last. But our hope is that we can figure out in the coming years, how can we make these TREs last longer or become permanent, and how can we reliably induce them based on every person's own individual presentation and symptomology. I think I have one final slide. And then, yes, so the next steps for this is we are starting to actually draft this paper. I've been working with the other members of the data team and the Renegade TRE5 for this. And we've got a tentative deadline of May of this year to get a paper describing these TREs up on medRxiv and submit it to some journal. So, put that in your calendars and keep your eyes peeled in the coming months for what will hopefully be one of the first real papers to come out of the Remission Biome Project. That's all I've got. Yes.
Okay. Hi, I'm Lisa, and I'm on the data analysis team, and I have MECFS and POTS. And for privacy reasons, I'm going to be keeping my camera turned off for this presentation. So I've been working on trying to gain insights into both remission events and general health for members of the R50 from a combination of wearable data and bearable data. And since MECFS is very individual, I've been focusing on searching for factors that make big changes for specific individuals. A lot of these factors might not show up in an analysis at the group level, but for specific individuals, they could make a big difference. And by uncovering patterns from individual data, we can gain insights into MECFS in general and remission events and the impact of various interventions on MECFS. And with that, here's a quick look at one of the things that we're looking into. So first, here's a graph of one of the R50's REM sleep. So the y-axis shows the duration of REM sleep they're getting as measured by their Oura ring on any given night, and the vertical black lines indicate the start and end of their remission event. So right at the beginning of their remission event, you can see that this person's REM sleep drops dramatically by over half. So there isn't a big change in their sleep duration at the same time. So for some reason, at the start of their remission event, their sleep architecture just changed, and it stayed changed for a period of time following the remission event as well. So one of the unique aspects of Remission Biome and Renegade research is that we're able to measure these changes every day in a realistic setting. Prior studies on ME/CFS and sleep typically rely on 1 or 2 nights in a sleep lab and only consider that data in the isolated study context. But using wearable data, we can track fine-grained changes over time and see how those changes relate to the overall context of other symptoms and interventions and just all of the other data we've collected. And at this point, we don't have enough data on remission events in general to say that REM sleep dropping during remission events is a consistent pattern. But anecdotally, we do have reports from a number of the R50 saying they tend to have lower REM sleep when they're feeling better. So overall, we're excited to explore what this means going forward and hope that it results in an increased understanding of ME/CFS, ultimately translating into an improvement in quality of life for patients. And with that, I'm going to hand it off to Sam.
My name is Sam Keating. I am a PhD student researcher at TTU-HSC, Texas Tech University Health Sciences Center, and I'm a big fan and advocate of decentralized patient-led research. As a published citizen scientist, systems biologist at heart, and ultimately, today I'll be talking about the preliminary findings we have from our Biomesight microbiome data. So on this slide, we have a view on the right of all 183 samples as stacked bar plots on the phylum level of all phyla per sample. So there's 183 samples that we have been able to process so far, more coming through the pipeline, and more that will fill in the gaps across different time spans of our analysis and our overall protocols. But what I wanted to highlight today is just an overview of some of the preliminary findings that have highlighted lactobacillus. So again, on the right is the phyla view of all 183 samples from various participants across separate dates. So an individual sample is a given participant's singular date sample, but participants, we aim to have multiple samples per person. So this is not clustered or sorted really for any time or other variable. It's just an overall view of just the complexity and diversity of the microbiome phyla involved. Whereas on the left, and we'll have a zoomed in plot on the next slide, is the genus level with different species. As you can see here, this zoomed in view is, again, lactobacillus. Both views are just lactobacillus genus and all the different species that compose it in our data set. So we also have a zoomed in view of the legend, which is specific to the species with different species making up a relative abundance of a given sample. So it's still all 189 samples from left to right, and there's some that are 0 or near 0. But what I wanted to highlight is this one outlier sample. You can sort of see it sticking up near the middle of the main plot towards the right, but on the left is a cropped view without zooming in of just how much it dwarfs the lactobacillus abundance compared to all other samples. And what's interesting as we follow the story of this singular sample is that this microbiome sample actually happens to relate to our TRE5 story that we've analyzed in more detail to date. So it's still early days for a lot of the microbiome analysis, but what's notable about this sample is that it's, once again, we uncovered, and we'll have future slides or upcoming slides on the nature in the TRE5 story. But again, on this slide, there is a diverse set of different species of lactobacillus, which are sorted alphabetically by color gradient within species names. And it's much more diverse than any known probiotic could explain. So even if there was something outside of the protocol or within the protocol that could relate to lactobacillus, it shouldn't really be able to explain the dynamics that we see here with this sample. So on the next slide, we have a zoomed in view looking instead of at just lactobacillus, looking at all of the sampling dates for just the TRE5 participants, which again is the transient remission event relevant participants. Of the 50 in the Renegade 50 cohort, having 5 with TRE events, my book is exciting, but again, still early days. And on the microbiome side, we have this shaded gray background for the TRE dates, for the start and end dates. And for the 3 that are wider than a singular date, this indicates that it's actually more than 1 day long of a TRE event. So a transient remission event that lasts more than 1 day, which is exciting. Even more exciting, we're able to start uncovering some patterns in the microbiome data where that sample that was highlighted earlier is actually right on the bottom left. And if we go to the next slide, there's a zoomed in view of this particular participant, the TRE 5th row participant, zoomed in on the left here. So that's that same sample I highlighted earlier. That color is lactobacillus. And the reason that there's so many different colors, the individual colors are at the genus level, and the individual bars or stacked bar elements that are in the same color actually represent different species of the same genus. So that's the way this R library called phyloseq organizes the plots I made. And what's exciting is we actually see this story is starting to unfold with a potential explanation of lactobacillus sort of exploding into dominance right before and sort of on the same date as the start of the TRE. The axis labels are small in this view, but they're actually within the same day. So we're investigating a little more closely, but between the different layers of data collection we have on the symptom bearable level, as well as the microbiome collection data, we actually believe that this is on the same date and not separate dates, but just with the way computational dates are stored, they can be within 24 hours and be labeled as separate dates. So we're still investigating and polishing some of that data, but for the several participants out of these 5 that have microbiome samples that are collected during or before and surrounding these TRE events, transient remission events, the TRE data is starting to highlight that lactobacillus might be a key marker of the entry into a TRE. And it's still early days to postulate whether that could expand a TRE to make it more reproducible or longer. But this is sort of the early days of some of the hypotheses we're able to generate from whole microbiome samples across many dates for the same participants. And the 183 samples includes a lot more than just these 5 that are highlighted in today's analysis and presentation on the microbiome data. But I just wanted to give a preview of what we have analyzed so far with these findings highlighting lactobacillus. And as a quick note, there's a few other bacterial taxa that are highlighted or looking promising as potential signals that are quite dramatic representations. So high percent abundance, which can even make up a significant chunk, upwards of 30% of a whole microbiome sample. Well, different numbers goes above 30 with the lactobacillus here. It's actually closer to 80% of a single sample. But there's a number that are in the 20 to 30-ish% range of a whole sample at a single bacterial taxa, like a single genus or even species. So we're starting to dig deeper into that to see what signals we might be able to uncover with respect to participants that had a transient remission event and those that didn't. But this is what I wanted to cover for today. And with that, I will pass it along.
Hello, everyone. So I am Kat Boniface in Riverside, California. We're not on fire currently, don't worry. I am a historian of science with a background in animal science. My primary trigger was a concussion in 2008, but my symptoms are incredibly similar to those here who have had a primary trigger of a virus or something else. So all roads sort of lead to ME for some form of metabolic immune dysfunction. And that, of course, is one of the things that we're trying to figure out here. I am one of the phase 1.5 participants, so I went through most of the Remission Biome protocol in May of 2023, and I did have Transient Remission Events, plural, as well as a significant baseline increase for that summer until I really overdid it in September and lost a lot of those gains, though not all of them. I'm also one of the one-to-one coaches. So this has given me sort of a unique opportunity to really dig into cases with people, looking at their symptom patterns, at the interventions they've tried, things that have worked, things that haven't. One of the things that I've been doing is looking at DNA. DNA testing was not required, but a lot of us have data. And whether it's a simple ancestry styles test or a whole genome test, there is still a lot of data to be gathered. There is an overrepresentation of certain gene variants. Some of these are expected, right? Like MTHFR is, fairly well known in the community to be something that affects a lot of us, but some of them were not. And these are not just risk factors. So there are hundreds, probably thousands of genes that add up to being potential risk factors if you, tip the balance one way or the other. Some of these, however, are actionable, and that's what I want to focus on today. In particular, we do see a higher incidence of glutamate hypofunction variants, mold detox variants. A lot of people have mentioned that this has been a significant issue for them, myself included, and multi-heterozygous metabolic disorders. I know that's sort of a mouthful. Basically, what I mean here is people who have heterozygous metabolic disorders, but lots of them, and often in the same area. So individually, they shouldn't have much effect, but when you start stacking them, it adds up. And these are particularly prevalent in participants who have been stuck, who have not been able to move forward, those who are either not stable enough or just have not been responding to certain treatments. We find that not only are these variants really common across our population, but they're even more common in these individuals. But once we identify them, solutions can be trialed. Next slide, please. So when does DNA matter? As we mentioned, DNA was not a requirement, but there have been a few people who didn't already have that data that I've suggested, maybe you want to look into it. Obviously, there are some reasons that people wouldn't want to do that, including privacy reasons. But here are some of the reasons to look at your DNA data, to get DNA data. Any individual who has a high incidence of adverse reactions to medications or supplements, or even dietary interventions, especially those treatments that have sort of a donut hole distribution among our population, where, a lot of people rave about it. It was the one thing that made a huge difference for them. It was great. And then a lot of other people were like, no, it was a disaster. And again, these can be medications, these can be supplements, and they can even be types of diet, ways of moving even, all sorts of things. Anything that has that donut hole response distribution, there's a very good chance there's a genetic reason. And that means there's a chance that we can find that reason and address it. Those who are very severe for an extended period of time. So a lot of us do hit very severe, but only briefly. Maybe we did a push crash cycle too many times in a row and we hit it, but we start to creep back up. Anybody who stays in that state, there's a good chance that there is a significant genetic burden there. As I mentioned, even within our population, these individuals do tend to have a higher rate of pathogenic genetic variants. And again, some of these are actionable, meaning we can do something about it. And the last one, and this might be a little more surprising, is a family history of autoimmunity. Okay, that's not surprising. Alzheimer's or schizophrenia specifically. These are closely tied to some of the actionable genes, which I'm going to talk about next. Next slide, please. So here are 3 categories of genes that are actionable. So one is glycogen storage disorders. So you may have heard Pompe is being studied in other contexts for ME/CFS because it can mimic it, but it can also exacerbate it. So it can either be that maybe it's just the genetic variant, and once you treat it, your symptoms are gone. That's rare, but it does happen. Maybe it is truly a misdiagnosis. More often, we have people who this is just something that adds to their overall symptom burden and prevents them from responding to other treatments. So as an example, McArdle's, which is a muscle glycogen storage disorder, has a diagnosable incidence worldwide. The estimate is about 1 in 100,000. There are at least 2 in Remission Biome that are diagnosable. The highest carrier rate estimate that I've seen for all pathogenic variants is about 1 in 160. And at least 10% of the R50 have one of these variants. That is a huge discrepancy. If it was just, a little bit off, okay, maybe it's just chance, but that is a huge discrepancy. Carnitine transport. And of course, a lot of us have responded well to certain carnitine supplements, so this is maybe not so surprising, but the prevalence of this is extreme. So this is the SLC22A genes. And in addition to simply carnitine, which is fatty acid metabolism as well as muscle metabolism, these genes are also important for metformin response, which is something that is, again, very commonly used in our community, whether as a prophylaxis or as a treatment. And then glutamate receptors. And those of you who have seen some of my talks before, the glutamate system is one of the things that I focus on. So I actually feel a little silly for not looking at the receptor genes earlier, because I was like, yeah, of course we're going to have lots of variants. We have tons of variants on the glutamate transporters, but there's nothing we can really do about them as, just by knowing the gene. So, But we have found that on some of the receptors, there are actionable variants. As an example, there is one particular variant, GRIN2A, which is an NMDA subtype. The minor allele is more common in people who are non-responders and in people who tend to have adverse reaction to a lot of the treatments. Again, those treatments that tend to have that donut hole distribution, because a lot of those treatments affect that receptor directly. So for a lot of us, we might need to tamp it down, whereas others, such as those with this particular variant, which is a loss of function variant, tamping it down is going to be a problem. That's going to create more symptoms. So again, once identified, treatments can then be adjusted. And last slide. So here are just a couple of examples. So among the glycogen storage disorders, there's a website that I've posted here that has fabulous resources for most of them. These are typically confirmed by either a blood test or biopsy. So anything that comes up in our genetics, it's just the recipe. It doesn't mean that we've got the cookies already. So we want to make sure that it matches our symptoms, it matches our labs. Some examples of interventions for McArdle's, the one that I carry: micropacing, right? So not just pacing your total activity, but within, even from moment to moment. A keto diet, for those that can hold it, is often used as treatment for McArdle's. And B6 is also a common intervention because it sort of boosts the activity of that particular enzyme. Within carnitine transport, this is again confirmed by blood tests. Fasting is contraindicated for these folks. They just cannot maintain their metabolism through fasting. Whereas for other people, fasting is beneficial. MCT oil and high-dose carnitine, there is actually a prescription version available, are also common treatments for carnitine transport disorders. Among the glutamate receptors, again, there's a website here with a lot of resources. Now, this website is really focused on much more severe variants, but that sort of gives us an idea of what happens when these particular genes are affected. So many of those on this website are ones that are not just loss of function, but absence of function, or in some cases, excess function. And so we can sort of look at the prevalence of certain symptoms at either end of that spectrum and see where we might fall. And that particular variant that I mentioned previously is more likely to have an adverse reaction to things that block NMDA, which of course are very, very common across our population. All right, so there are absolutely a ton of these. I am happy to talk to anybody about them if you either have these variants or are curious about them. But for now, I will pass it on.
Hi, everyone. I'm Tess, co-founder of Remission Biome. And I had a life-changing transient remission event in 2009. I went from nearly bedbound to mild for about a decade afterwards. So I'm really excited to see where Remission Biome has gone. You know, this originally started off as just the question, can we recreate the transient remission events? And honestly, I didn't think that we were going to be able to do it. I thought it was just this rare event. And so I was surprised when we got into the pilot and we were actually able to do it. I was another one that had these transient remission events. And I had multiple throughout the process. And then watching the 1.5, the people that reached out after the pilot and seeing one after another having some of these remission events was really amazing. So I'm going to talk a little bit about just overall, what have we seen with Remission Biome? We get this a question a lot, you know, where are you guys in the process? How many people have had these transient remission events that have tried the Remission Biome protocol? And so some of this is based on a post-acute survey that we sent out to people who went through the antibiotics phase. For the top here, I just want to mention these are self-reported and they're subjective, and you know, Sam shows some clues. You know, we might be able to start getting objective measurements of it. Maybe in the next year, we'll be able to actually, you know, be able to tell when a remission event happened just by looking at objective data. But for now, caveat, it's self-reported. The N is small so far of how many people have gone through the antibiotics phase, so we can't really draw clear conclusions from this. I just wanted to show you this data just so you have an idea where we are and how many people have done this. So for the pilot, 2 out of 3 people had these dramatic full remission events. For the 1.5 people that reached out, it was 9 out of 10. And then for the Renegade 50 so far, it's 1 out of 10. So total, adding those all up, about 60% of the people who tried the protocol and took the antibiotics had these transient remission events. Another question we had was, what happens longer term? I mentioned that I had this increase in baseline in, you know, decrease of my symptoms after my first remission event in 2009 for a really long time. So that was a question we had from the beginning. You know, what happens afterwards? If you have these transient remission events, does that lead to a longer term increase in your baseline? So what you see below is, again, based on these post-acute surveys, just asking people how they did after they went through the acute phase. And again, this isn't very many people. 15 people filled out the survey. So again, we can't really draw clear conclusions from this. And yeah, I mentioned, you know, 6 out of 21 participants, they didn't fill out the survey. So we're not sure how they've done since they did the antibiotics. But interestingly, and I'll talk a bit more about these baseline increases. One of the questions was, do you need to have one of these transient remission events to have a baseline increase? And we found that the answer was no. Some people did not have a TRE, but they did have a baseline increase. Some people had these remission events and afterwards they felt better longer term. Others had no change though afterwards. So one thing you can tell from this, I said we can't draw any conclusions, but it's just that it's not consistent. You know, it's really individual, what might happen afterwards. Next slide. Okay, so I mentioned looking more closely at the baseline changes. Again, for these ones, when we first started the pilot and the 1.5, we didn't really have as much of the formalized study that we have now. So the people weren't filling out validated questionnaires. And so a lot of this that I'm showing it's self-reported, it's from memory, it's, you know, coming back to people afterwards and saying, what happened with your baseline after you went through this phase? So since then, with Renegade 50, we are having people do validated questionnaires. And so as we move forward, we'll have better data. One of them is the Bell score. And so rather than just showing, you know, did you have a baseline decrease or increase, we'll be able to get more detailed numbers about that. So another caveat, 3 of the participants who filled out the survey aren't more than 9 months out, so they aren't included in those figures. But looking at that, some of the things stand out. Some some people did have a baseline decrease right afterwards. We didn't know if that was going to happen. But looking forward, it's possible that they longer term go back to where they were before the antibiotics, or maybe even have an increase compared to before they did it. But there's just a lot of open questions here. We're just getting started, and no conclusions from this. But now you can see, you know, how many people have gone through this, what we've captured so far. So that is it. Go to Isabel.
All right, so a little bit about what we have learned here. To summarize it, we know that we can trigger remission events. So we did that. They have occurred in 60% of the participants. We have also learned that antibiotics aren't always required to have a remission event. And we'll be talking later down the line as to what other ways and what other interventions can cause this to happen. So in terms of the physiological mechanisms of transient remission events, like Tess said, we still have a lot to learn. And we're looking at those clues, right? That have immune, metabolic, the microbiome, sleep, all of the data that it's being collected. There could be genetic differences on non-responders versus responders in terms of the remission events, but also in terms of the baseline increases and the interventions being customized to those differences. And we need to understand the mechanisms that lead to long-term or even permanent remission, which is the ultimate desire. So long-term health challenges, 50% of people who have done the Remission Biome protocol have had baseline increases at 12 months, and the antibiotics are not required for that in a very small percentage of people, right? And we have massive data sets that can be analyzed later on for a broader question. Like Elly mentioned earlier, in February, we had people add a lot more of the separate interventions that they take and the symptoms that were not required to be reported. So all of that can be analyzed at scale at a later time. Right now, we have just gained access to a great tool to be able to do that. The name of the platform is Quinn, but it is still too early to tell if that will give us the outputs that we need to see to look at all of these correlations of all of the data that we have gathered. Next slide, please. Okay, so what are we doing next? We're doing additional testing and data analysis, like we said, for the R50. We're getting ready for the next cohort. And in this cohort, because of the things that we have learned here and how valuable the completeness of that data is, we are aiming to do what we call moonshot testing. So in that phase 1, the testing will be so much more robust. So the data that we gather in order to understand those individual participants at a much deeper level will also be more robust. Higher number of participants, more standardization of the data when it comes to wearables, the things that are inputted in Bearable, again, the testing that it's done before, during, and after, and the times frames for those data to be collected. And also continue to do the analysis of the factors that are outside of the protocol in itself. So as you guys know, when a clinical trial is run, people may not respond, but there is no further investigation as to why those responses didn't occur. Because the data that is collected is very limited and very narrowly focused on what is being studied. We want to understand things, for example, if somebody has CCI, which we encourage everyone to test for if it is possible in stage 1, how does that interfere with the progression of the their baseline increases if there is no surgery. We also have reported people who have decreased in symptoms of CCI during transient remission events, that it's very worth investigating what the mechanisms of that is. So many different ways to go with this. We're also going to be testing rutin, which is one of the interventions that has led to remission events in some people. Additional projects that complement our understanding of all of the dysfunction that happens within ME, studying the glutamate system with academic partnerships, and more traditional placebo control trials in the future as well. And that is all from me. Next.
Thanks, Isabel. I hope everyone is excited as we are about what's possible. In the beginning of our presentation, I showed how much data we're gathering, hundreds of thousands of data points. And what each of our presenters showed today is just scratching the surface. And also, how I mentioned at the beginning, our biggest challenge on the data analysis team is that we have limited spoons. So we're moving slower than we'd like to. Our energy levels can fluctuate dramatically, and that makes it hard to gather, anonymize, explore data, let alone find patterns and synthesize hypotheses. So we're always seeking help from the community. If you have experience with data analysis and can volunteer some time, please reach out to me at mike@renegade-research.org. And with that, we'll be opening it up for discussion and for Q&A.
It looks like we have a question in the Q&A from Athena Hall, and I will bring her on so she can ask her question live, if you're ready for that, Athena.
Thank you for your incredible hours of dedication and your brilliant work. I just wanted to ask, we all know that biomechanical issues now play a larger role, and they may not be unstable patients or tethered cord that far along. However, I want to say that they can be persistently misaligned and can be generating as a consequence significant symptomatology which ME patients experience. So there can be a really big overlap here. How do you manage this complication, because it is such a significant complication with the lack of appropriate imaging as we know it? For example, limited dynamic imaging that we have before, against patients become fully unstable. They can be partially unstable or not unstable at all and persistently misaligned, which can result in further problems. So how do you manage this complication? Because it really is a difficult job.
So this is not quite about managing the complication, because that's not my my
I should say
area.
analyze. How do you integrate that in the analysis is what I'm trying to ask.
So one of the things that I've been really excited about is how some of these craniocervical issues do appear in the symptom co-occurrence networks. We do get little modules in that network that we interpret as showing signs of mechanical issues. So that would be things like neck pain, shoulder pain, certain craniocervical junction issues. And so, you know, that's not something we're necessarily conditioning on, but I think we are seeing a pretty robust signal of it in the data itself. And hopefully that will become part of this larger, you know, push to make mechanical basis issues like something that the scientific community takes more seriously.
It's a logical standpoint you take, and I want to say that one of the challenges in the CCJ arena patients is that they don't all present the same way. So for example, some may have cluster headaches as a consequence of being misaligned. Others may have POTS or mast cell. And I want to say that it would be fair to say that it could be one of the top 2 driving factors in this big problem. So I'm just drawing your attention to the fact that more information is now available, but imaging still continues to be inconsistent, and technicians are not properly trained from area to area. Even if you do have access to an upright MRI, it doesn't mean you're going to have appropriate imaging done. So it's a super challenge to consider in the analysis. So I'm just drawing your attention to that piece.
Yeah, and I'll add a little bit to that because the challenge is really significant. We do everything that we can to encourage participants to seek diagnosis in the first stage of the protocol, but that's not always possible, especially for international participants. There is more movement in awareness and integrating more clinicians, and the part of the education that we do is towards that aim, because we need to expand the pool of clinicians who understand this condition, who can order appropriate imaging, and who can take action after that. So we have those resources to share with the participants and do the practical advocacy to help them connect wherever possible, but that remains a huge obstacle and a huge challenge. We hope in the future to take on projects that expand on the availability of the diagnoses, things like mobile imaging. I don't know if you know that Dr. Gillette, for example, does CBCT as opposed to, a full MRI, a regular MRI, which can be extremely helpful if we were able to do that here. So we're looking towards the future and being able to integrate more things into the overall picture.
Thank you. I was going to mention that you actually brought Dr. Gillette's group and Amy, who discussed the possibility of CBCT. Not everyone's in agreement on it, but regardless, they've developed protocols which are credible, and it provides an option for analysis, which can require, as very cost effective and also very tolerable for patients who are in pain. And then I want to say, just as a suggestion, that when we look back longitudinally based on Dr. Rowe's work, we may have our analysis and your work, and then years later, we may have a patient that presents with stenosis or something that has become more significant. And then we can look back historically, like a post-analysis, which will give us a different perspective and conclusion. So we're all working on this together.
Yeah, I'll add a little bit to that. We obviously think that the mechanical issues are really important because we're going to be we've had 2 roundtables so far, which are on a YouTube channel, and we're going to be having a 3rd one. I believe it's next week. Dr. Navio is going to come and make some of the connections between the mechanical issues and molecular issues. And then I also wanted to add something that's fascinating to me. Many of us who've had the transient remission events have mechanical issues. So I'm one of them. I have many of the issues. I have tethered cord. I have CCI. I have had surgeries for a styloidectomy. I have all the things. And I was still able to have these remission events. And so to me, that's brings up a really interesting question about the pathophysiology of ME/CFS, because I know that these issues are important, but you can also have the remission events.
Yeah, of course. Thank you for your incredible work and your thinking and your critical thinking and your analysis as a group. You're brilliant.
And just one other piece on that is a cause or as a reaction to, because as you get inflammatory processes and pathways going on, they can break down cartilage and bone, and so it could be it can be cause and effect, and certainly the end state of fibrosis is an important piece, which wouldn't necessarily be in cranial instability, but that just for always remembering the cause and effect.
I'd like to share that one of our most recent TRE people asked the question is, can I just have a remission event that's just related to all of my neck stuff? Does that count? And we had to reassure her, yes, it counts. If she did have the complete relief of fatigue for 3 hours, but the relief from all of her neck stuff is going on for weeks now. So this is a very special thing. We are encouraging the people that have this relief in their neck symptoms to, track those symptoms carefully, or the lack thereof, make sure that you're not carrying it over. Maybe add different things into your symptom tracking. As stuff comes back, maybe it feels a little different. We are trying to capture it, and Thomas is capturing it in the symptom clustering. It's an exciting thing, and I think John has got a point there. You get somebody, their inflammation down, and all of a sudden things work differently, and you feel better. And you go back to maybe you may have always had this for your whole life, but you didn't feel these symptoms until everything else stopped working as efficiently.
Thank you.
Our next question that is in the Q&A is from Rivka, and I will go ahead and bring you up if you're ready.
Hi, I have no idea what question. I'm putting so many questions in. One thought I had is, of course, we all know that antibiotics can have antiviral properties. I'm just wondering if the one that you all are finding so effective has that. And then the other thing I wanted to say is that Martha Ecky's massive survey results could be really useful for deciding on what treatments. Thank you all. It's just so exciting.
I would say anecdotally, something that helped me a lot was working on the microclots at the same time as inflammation for long COVID, and that came very specifically out of the Martha's study, her original one, of that the kinases made a difference.
Yeah, and I was going to add to that I mean, we don't prescribe obviously any treatment. This is something that the participants do in conjunction with their providers, right? And we know that many people are non-responders to one treatment, when others it's incredibly helpful. So to have that aggregation in the survey is very useful for participants to have that conversation with their providers. And what we're doing within the context of the project is, like we shared earlier, having participants add all of the treatments and all of the symptoms that they are experiencing, so that can be also later analyzed as a whole. So if we find that, let's say within the protocol, just throwing out words here, 5 people didn't respond at all, they didn't have TREs, and they didn't have baseline increases, and those 5 people are also taking LDN and montelukast. Okay, so that gives us an insight that's outside within the parameters of the project to look at what are the commonalities of all of these treatments, symptoms, genetics, hormones, microbiome, when you aggregate all together, and what makes sense when they do get analyzed. Are there patterns? Are there signals? Can we find something really useful there?
That makes a lot of sense. I think that, if we could figure out subsets of groups in ME/CFS, I'm just thinking of the rituximab study. The publication said basically this isn't effective, but if we had only known what subset of patients would have found it effective. Yeah, anyway, thank you all very much.
One thing I could point out in that regard is sort of the power, or one of the strengths of our data analysis and what we collected so far that we have not finished yet as far as data analysis, is just how synergistic the different data types are across the joinable time points. So for the same participants, same time points, or over time for the same participants, we can connect these different data types that might allow us to do some subtyping development. So hopefully we can actually comment on that in the future.
I think there's only one more question in the Q&A from an anonymous, and antibiotics can also be very anti-inflammatory. Is this significant? Who wants to take that one?
I'm happy to give a shot at it. So if you think of the dimensions of inflammation, antibiotics, so quieting the bacteria or killing the bacteria, and antiviral, same on the virus of stopping the replication. There's a lot of overlap in pathways, especially with the things called the toll-like receptors and some others that are, you can have multiple things turning them on that can have as an endpoint an increase in inflammation. So I think we're at the early stages of really understanding all that complexity. But just as one specific example, then I'll hand it off. Lyme does TLR2, and the spike and lipopolysaccharide and some other things do TLR4, but they both have a common pathway in terms of how they light up the innate immune system. So if you have one, you would expect it to be more causative on the other. Let's say you had chronic Lyme and then you got COVID and had some spike hanging out, you would expect it would amplify.
Thanks, John. I think we are out of questions. Anything else anybody wants to mention?
Yeah, I just wanted to add, I forgot to mention the single cell immune profiling results that we got. So we didn't go into that today because there was a whole roundtable on it. And so we'll send that to everyone. It looks like Rochelle just did. So that's another really, really fascinating result that we've found. And you might, if anybody's interested, check out that video. There were major differences in my pilot data during the antibiotics and for months afterwards.
All right. Wonderful. Thank you.
Oh, just real quick, one of the questions had come in about can you use a Fitbit? And the answer is yes. Right now, we don't have a standard wearable device that everybody uses. We encourage everybody in the program who has a wearable to wear it as much as possible. But that's something that we're looking at for the future, perhaps standardizing on one particular device. So we're looking at that for the future.
In some devices, you have to sync more often than others so that your data is kept. So that's
right.
a consideration. For people who have an old Apple Watch and a new Apple Watch, you can keep them both charged and keep them both working for you so that you don't miss that time of battery charging. Wearables are quite interesting and varied amongst us right now. And reminder that if you want to know the details of the protocol in 2 weeks on a Friday, February 7th, John will be explaining the protocol in the clinicians roundtable. So next week is Dr. Navio, and 2 weeks is John and the protocol. And then a 2-part series starts with Dr. Rowe and Dr. Isola, clinical and research update.
Basically, you guys just want to reserve all of your Fridays at 1 o'clock to hang out with us. Occasionally Saturdays, but basically all of your Fridays. Come talk to us. Yes, 1 o'clock Eastern. Thank you, Kat.
Hey, it looks like we got one last question about HEPA filtration and air quality, and it's just such an important piece. Yeah, it is a, it's fairly low cost and certainly low morbidity. And if there is mold or there in the spring, if there's pollen or any of these things can jack up the immune system. And so give it a try and you may notice a change for sure.
Yes, there is one event on a Saturday. They're regularly Friday. Which is the date of that?
It's Saturday, February 1st is the first Hopkins one. And then the second one is February 14th. I think I misspoke earlier. Another thing you can do in winter that's a low-hanging fruit is make sure you keep your space humidified if you're in a place where you have to keep the heat running all the time. Just one less stress on the body to stay hydrated.
All right, everyone, we're going on to an hour and a half. So in the name of spoons, we're gonna end it here. You can find us on X at Renegade Res, and send us questions or let us know anything else. Thank you, everyone, and have a wonderful rest of your day and weekend. Bye-bye.
Thank you.